One of the very first spiders to capture my attention as a child were these large, black, velvety creatures that built messy retreats under the eaves of my back porch. Something about these little animals intrigued me and made me want to know more about them. Even nearly 2 decades later, Kukulcania hibernalis captivates me whenever I encounter it. So today we're going to take a look at this interesting animal and try to understand its place in the world of spiders. So join me as we explore life in endless forms most beautiful. I didn't know anything about evolution at the time, but I did recognize certain traits about this particular species that reminded me of tarantulas. At least what I had seen of them in books. It would be years before I had the opportunity to see one in person. I couldn't articulate exactly what it was that made this spider stand out from more typical spiders, but something about them just stood out to me as distinct from other araneomorphs. Its silky black hairs reminded me of a miniature version of the tarantula Pamphobeteus antinus. Its broad and prominent coccyx reminded me of the pictures I had seen in my favorite childhood books of large megalomorphs viewed ventrally. It also has long pedipalps that look more like a fifth pair of legs than the highly modified structures of more derived spiders. But what stood out to me most was a unique attribute of its life cycle. Unlike other araneomorphae, and very much like more basal spiders, females continue to molt after reaching maturity and can live for many years beyond their maturing molt. And this is actually the reason for one of its many colloquial names, the southern hibernating spider. So named because it lives year to year rather than dying off each winter and hatching in spring as many others do. When I was first starting out in the invertebrate hobby, I found that I had a desire to understand my favorite species in their broader context. For example, I knew that tarantulas were megalomorphs, but I didn't really know what this meant. I knew it involved the possession of certain traits, but I didn't understand why they have those traits or where they come from. I found the subject of arthropod taxonomy daunting, full of huge words that defy pronunciation, and the need for a lot of background knowledge on a variety of subjects that I would need to familiarize myself with before any of it would really make sense. I suspect this is the case for many people who enjoy the wonderful world of arthropods. They would like to appreciate them in the broader sense, and see where their favorite animals fit into the grand scheme of life, but the subject can be very dense if one approaches it with little familiarity. For this reason, I will include such context in my overviews of groups and species as best I can. This way, by exploring the diversity of the group, in this case spiders, we will also be exploring their evolution and taxonomy as well. As it happens, the group I want to talk about today can be seen from near the base of the spider family tree. So let's take a quick look at the root of the spider tree. I'll do a more in-depth overview in a later entry. We don't need to go very deep into the spider tree because our subject is very primitive compared to more derived species and is found near the root of the araneomorphs. So to start, spiders are arachnids, which are a subset of a group called chelicerates, a branch of arthropods emerging from the parent group during the mid-Cambrian about 500 million years ago. As a group, chelicerates represent a more basal arthropod form than, say, insects. The exact relationships among arthropods is a complex web to untangle, since they are the single most diverse branch of animals to have ever existed, collectively dwarfing the entire rest of the tree of life put together. Molecular data is being used to establish a higher resolution look at the relationships among them, but that's a story for another time. Spiders represent a subgrouping of arachnids that form the clade RNA, or spiders. From here, the tree branches again into two groups representing the totality of what are considered to be true spiders. The Opisthothelae, which includes virtually all extant spiders, and the Mesothelea, representing many extinct spiders and only one extant family, the Filistatidae. The name Mesothelea means middle nipple, named for their spinnerets being placed near the middle of the underside of the abdomen. Opisthothelae means backward or behind nipple, placing the spinnerets at the back of the abdomen as in most modern spiders. The Opisthothelae are further divided into two broad groups, the Megalomorphae, including tarantulas and trapdoor spiders, and the Araneomorphae, which is all the rest. Megalomorph spiders retain a variety of features that are basal to all spiders, including the full set of 4 book lungs, vertically operating chelicerae, And they have lost some other features, most notably the complete loss of the anterior median pair of spinnerets, as well as the visibly segmented abdomen present in most arachnids, including basal spiders. So here's a visual illustrating the abdominal segmentation typical of arachnids. In the top left, we have a Lephistiid spider, the only living representative of Mesothelea. On the right, we have an Amblypygid. On the bottom right, we have a Thelephonid, also known as a Vingaroon. Very cool animal. And on the bottom left, we have, of course, a scorpion, which is probably my very favorite of all arthropod groups. Most araneomorphs have lost one set of book lungs. While many retain the anterior pair, they now feature a modified posterior pair as a trachea. Their fangs cross each other in a pinching motion, and they retain the anterior median spinnerets, or at least a vestige of it. More on this in a moment. The next branch is where our subject can be seen on the horizon. Traditionally, araneomorph spiders have been split into two main groups, the Intelliginae, which possess a hardened or sclerotized female epigyne, and the more basal Haploginae, which lacks these more complex reproductive structures. A third clade, the Hypochilidae, are sister to all other araneomorphs and represent the most basal of all extant araneomorph spiders. The vast majority of extant spiders are members of the Intelliginae. However, in the age of molecular data, the grouping Haploginae has been shown to be paraphyletic, meaning that it included a common ancestor for most, but not all, haplogyne spiders. That is to say that no common ancestor could be identified that included all haplogyne spiders exclusively. This means that rather than two subdivisions of Araneomorphae, the haplogyne side of the fork has been split into several groups to better represent the true relationships of extant spiders. Molecular studies do indicate that the Haploginae group did include a monophyletic clade, but that a few interesting outliers were lumped in with it, making the taxon paraphyletic. Most of the traditional haplogynes were found to form a clade and are now referred to as the Synspermiata, the collection of Cribellate haplogynes. One family, the Leptonetidae, was shown to be likely basal to the Intelliginae, although this is tentative. More research will be needed to resolve these relationships in greater detail. One family previously grouped among the haplogynes, the Filistatidae, was revealed to be a sister group to the Hypochilidae, the so-called Paleocribellate spiders. This means that Filistatids are among the oldest and most primitive of araneomorph lineages. The Hypochilidae Filistatidae group diverged from the rest of the spiders during the mid to late Mesozoic era, somewhere between 120 and 294 million years ago, and represents one of the earliest developments in araneomorph history. The Hypochilidae represent the most basal of extant araneomorph spiders. They possess the defining intersecting fangs, but still have 4 book lungs like megalomorphs do. The Filistatidae is what we are interested in today. Filistatid spiders are collectively known as crevice weavers. Defining characteristics of the group include the retention of the cribellum, a structure derived from the anterior median spinnerets present in primitive spiders, strong sexual dimorphism in mature individuals, the retention of all segments on the anterior lateral spinnerets, and the vestiges of the anterior book lungs during development. The cribellum is a silk-spinning apparatus present in some Araneomorphae, usually more basal clades. Spiders that retain this feature are referred to as cribellate spiders. Those lacking it are said to be ecribellate. Most araneomorphs are ecribellate, but many still retain a visible vestige of the structure called a cololus. The retention of the cribellum is one of the features that distinguishes the Filistatidae Hypochilidae clade from Synspermiata, which are a collection of ecribellate haplogynes. The cribellum is made of one or more plates with small holes or perforations. Spiders that retain a cribellum also feature a comb-like row of hairs on the hind legs called a calamastrum. This is used to tease out the non-adhesive silk for the animal to use. The result is a mess of extremely fine silk strands. This unique type of spider silk is called cribellate silk. Because cribellate silk is not adhesive, the mechanism of prey capture relies on entanglement. Cribellate silk is typically wadded such that it can be stretched and unraveled when agitated. The more a prey item struggles, the more entangled they become. Some examples of cribellate spiders of other taxons include the ogre-faced Dinopus, the industrious Agellanopsis, a common feature in many backyards, the beautiful Erisis, and the fascinating and enigmatic Ulyboridae, notable not only for being cribellate spiders that produce orbs, which are an advanced form of trapping generally seen in more derived spiders, but they are also entirely non-venomous, having lost their venom glands at some point during their evolution. Filistatidae and its sister clade Hypochilidae do something unique with their cribellate silk. they fold it. Cribellate silk is already messy, but this folding tends to create a very distinctive look for the silken structures of the group. K. hibernalis is one of the most evolutionarily significant spider species you are likely to find in your yard. Common in dark, dry places like sheds, wood piles, underneath porch covers, this species is easily spotted from afar by its distinctively messy webbing. This species also has an enormous distribution, being widely distributed throughout the southeastern United States, Central America, most of South America, and even one confirmed established population in West Africa. They tend to prefer areas that are not often disturbed. I located most of the specimens featured in this video in an old pump room housing a colony several generations old, in undisturbed wire trays, and among the items on an old shelf. As a child, I frequently found them under porches, in old sheds, and around conduit and plumbing entering old buildings. You can see from the images that they are capable of modifying their retreats depending on the opportunity. Sometimes they create neat holes, sometimes messy conglomerations. They can be oriented vertically, horizontally, or even entirely upside down. Most of the specimens I encountered displayed a strong preference for verticality. When possible, they will create a retreat into an opening and surround it with silk. However, they can prove very resourceful if this ideal cover is not available. They can use corners or even make use of random debris or the remains of old prey in order to fashion a retreat out of whatever is available. I remember as a child I used to love sneaking out onto my back porch at night and see the large females just outside their silken retreat waiting for food or mates. These animals are easily startled, often retreating at the very first sign of danger, but they are also easily teased out with a little movement. K. hibernalis displays strong sexual dimorphism common to Filistatids. Upon maturing, males take on an entirely different color. Interestingly, the male of this species bears a remarkable resemblance to recluse spiders, Loxosceles species. This spider is responsible for many false reports of introduced Loxosceles populations due to mistaken identity. If we compare the mature male K. hibernalis on the left to the L. reclusa female on the right, you can note that K. hibernalis has a total of 8 eyes clustered densely together at the front of its proxima, while Loxosceles species have 6 eyes spread out symmetrically in pairs. More noticeable are the long pedipalps of K. hibernalis compared to Loxosceles, and the lack of a violin shape on the carapace. As an interesting side note, recluse spiders are a part of Synspermiata, being members of the family Sicariidae. Like Filistatids, they were once grouped among the so-called haplogyne spiders, but do not retain a cribellum and only have 6 eyes. Female K. hibernalis continue to molt regularly even after reaching maturity, a trait common in megalomorphs but rare among the uraniomorphs. Another interesting feature of the Filistatidae is the temporary appearance of the posterior book lungs in early instars. I have included in the description a link to a study in which you can actually see a visual of this taken by scanning electron microscope. These spiders also seem to be remarkably tolerant of each other. Adults and juveniles can often be found clustered together in dense populations. Even large females guarding an egg sac can be found just inches away from other spiders. Some research has been done that suggests that, at least as juveniles, this species could be considered borderline social, with the individuals becoming more territorial as they grow. Regardless of the degree of social behavior they exhibit, they do seem to be densely concentrated where they do occur, with populations occurring in clusters, with several generations sharing the same living space inside of old sheds and familiar places. One last interesting thing to look at is the mating behavior of the species. The mating ritual takes a bit of time. The male spends a while announcing his presence, trying to draw the female out without becoming dinner. The mating process does bear some resemblance to megalomorphs as well. If I am able, I will make another video documenting the mating behavior of Filistatids in the future. Overall, Kukulcania hibernalis is an interesting spider. Its early divergence makes it a classic choice for studies of phylogenetics and comparative morphology. This relic of evolution and of my childhood will always capture my attention whenever I encounter it. So the next time you find a big black spider in a messy web, before you reach for the bug spray, take another look. You may just be encountering something special. Thank you for watching. If you enjoy content like this, please consider subscribing and be sure to hit the notification bell to be alerted when new content is available. Also, check out the links to the papers included in the description if you are so inclined. Thank you, and see you next time.